Hi, we're Team RibOxSwitch. We're a group of students from Oxford, and we've been building tunable cooperative genetic switches out of RNA. Come and see what we've been up to. All living things are made of cells, and each cell in your body contains an identical set of genes. Genes are long sequences of DNA, many of which are instructions for making proteins, the complex molecules that accomplish all sorts of important tasks, such as catalyzing chemical reactions. Although all your cells contain the same genes, the cells found in your brain look and function very differently to those that are found in your heart. How do cells decide what to do? And how do we make them do what we want? It all comes down to gene regulation. Cells can turn genes on and off, regulating the types and amounts of proteins they produce over time. When a cell needs to make a protein, a polymerase transcribes a copy of the relevant DNA, producing single-stranded mRNA. A molecular machine called the ribosome engages at the ribosome binding site. Then it reads the mRNA, and according to the order of bases, it attaches amino acids together to make the protein. In a cell, the pattern of gene regulation is very complex, as many genes are expressed and regulated simultaneously. Changes in the rate of production of specific proteins affects their abundance in the cell, which gives rise to differences in cell appearance and behavior. Telling cells which proteins to produce and when to produce them has vast applications in medicine, research, and biotechnology. So we decided to make a genetic switch that can turn a gene on or off in response to the concentration of an input molecule. Our switch works on the level of mRNA. It affects the ribosome's ability to start protein production, and as such, it's called a riboswitch. Our riboswitch is a stem loop structure in front of a gene. In the off state, the stem hides the ribosome binding site, which prevents the ribosome from engaging. Since it can no longer read its instructions, the production of the protein is turned off. Short RNA strands that we call triggers can bind to their complementary binding sites in the loop of the switch. Trigger binding destabilizes the stem and opens the switch, making the ribosome binding site accessible. This means that the production of proteins can begin. The gene has been turned on. We can alter the behavior of the switch by changing certain aspects of its design. By changing the length of the stem, we should be able to tune when our switch turns on and off. By inserting more identical binding sites into the loop, we can make the transition between the on and off states sharper. This is a cooperative mechanism. The first trigger that binds makes it much easier for the other triggers to bind, accelerating the opening of the switch. Having binary on-off behavior is useful in systems when you want to make clear decisions. Our design is modular. The switch could be used in front of any gene. The triggers will only bind to the loop sequence and open the switch to which they are complementary. Multiple switches could be controlled independently by choosing distinct loop binding sequences. We've also designed a repressive switch that works in a similar way but in reverse, turning protein production off rather than on. Integrating our rationally designed riboswitches in genetic networks, we'd hope to create and control complex patterns of gene regulation. We can imagine our switches tuned to specific concentrations of pollutants or pathogenic markers, or employed to self-regulate metabolic processes in industrial biotechnology. If you'd like to learn more, head to our team's website. Thanks to all our supporters, and thank you for watching.